Welcome to Seed School India. In today's video, we will explore the fascinating role of PEX proteins during seed germination. Although these proteins are not widely known, they are essential for building functional peroxisomes, which help convert stored fats into energy, regulate reactive oxygen species, and support healthy seedlings growth. By the end of this video, you will understand how PEX proteins contribute to successful germination and why they are important in modern seed biology and crop improvement. Seed germination is the process through which a dormant seed develops into a young seedling. It begins with water absorption, known as imbibition, which activates enzymes and restarts metabolic processes. The stored food reserve inside the seed are then mobilized to provide energy for growth. Finally, the radical emerges and forms the primary root. Successful germination is the first step toward healthy crop establishment and is strongly influenced by efficient cellular metabolism. Peroxisomes are small membrane-bound organelles found in almost all plant cells. They perform several important metabolic functions, including the breakdown of fatty acids, detoxification of hydrogen peroxide, participation in the glycolate cycle, and metabolism of certain plant hormones. During seed germination, especially in oil-rich seeds such as soybean, sunflower, and castor, peroxisomes become highly active because they supply the energy required for early seedling development. PEX proteins, also called peroxins, are specialized proteins responsible for the formation and maintenance of peroxisomes. They control how peroxisomes are assembled, how enzymes are transported into them, and how these organelles divide and function properly. Scientists have identified many PEX genes in plants and other organisms. Without these proteins, peroxisomes cannot perform their normal metabolic activities, making them indispensable during seed germination. PEX proteins ensure that peroxisomes receive all enzymes required for their metabolic functions. When PEX proteins are defective, enzymes remain in the cytoplasm instead of entering the peroxisomes. As a result, fatty acid breakdown slows down, energy production decreases, and seedling develop poorly. Studies in Arabidopsis have shown that mutations in PX genes often lead to delayed germination and weak seedling establishment, highlighting their critical biological role. Peroxisome biogenesis refers to the formation of new peroxisomes inside plant cells. This process begins with the membrane assembly, followed by the insertion of membrane proteins and the import of metabolic enzymes into the organelles. Key PEX proteins such as PEX3, PEX16, and PEX19 coordinate these events. Once mature, peroxisomes multiply and become fully functional, allowing germinating seeds to efficiently mobilize stored food reserve. Most enzymes required inside peroxisomes are produced in the cytoplasm and must be transported into organelles. This transport occurs through two major pathways called PTS1 and PTS2. PX5 recognizes proteins carrying the PTS1 signal, while PX7 recognizes proteins carrying the PTS2 signal. Together with docking proteins such as PX13 and PX14, they ensure that the correct enzymes reach the peroxisomes at the right time. PX5 is one of the most important peroxisomes because it functions as a transport receptor. It identifies proteins carrying the PTS1 targeting signal in the cytoplasm, binds to them and delivers them into the peroxisome. After releasing its cargo, PX5 returns to the cytoplasm to repeat the processes. This recycling mechanism allows efficient import of many enzymes during the rapid metabolic activity associated with seed germination. While PEX5 recognizes PTS1 proteins, PEX7 is responsible for transporting proteins that contain the PTS2 targeting sequence. Many enzymes involved in fatty acid metabolism depend on this pathway. PX7 often works together with PX5 to complete the import process. This coordinated transport system ensures that peroxisomes contain all the enzymes necessary for lipid breakdown, energy production, and successful seedling establishment. After delivering enzymes into peroxisome, PX5 does not remain inside the organelle. Instead, it is recycled back to the cytoplasm so it can transport additional proteins. This recycling process involves proteins such as PX1, PX6, and PX26, which use cellular energy to remove PX5 from the peroxismal membrane. Efficient recycling ensures continuous protein import and keeps the peroxisome fully functional during the active phase of seed germination. Many economically important crops, including soybean, sunflower, mustard, groundnut, and castor, store most of their food reserves as oils in specialized structure called oil bodies. During germination, these stored lipids serve as the primary energy source because photosynthesis has not yet begun. The stored oils must first be converted into usable energy and sugars, making peroxisomes and PEX proteins essential for successful seedling growth. The first major step in utilizing stored oils in B-oxidation, which takes place inside peroxisomes. In this process, fatty acids are broken down into acetyl-CoA molecules while releasing reducing power for cellular metabolism. Acetyl-CoA becomes the starting material for further metabolic pathways that generate carbohydrates and energy. Without properly functioning PX proteins, the enzymes responsible for B oxidation cannot enter the peroxisome, severely reducing energy production. The glyoxylate cycle is specialized metabolic pathway found in germinating oil-rich seeds. It converts acetyl-CoA produced during B-oxidation into compounds that are eventually transformed into sugars. Key enzymes such as isocitrate, lyase, and malate synthase makes this process possible. These sugars provide the energy and carbon skeletons needed for cell division, tissue development, and early seedling growth before photosynthesis becomes active. The conversion of stored oils into carbohydrates follows a well-organized sequence. First, triglycerides are broken down into fatty acids. These fatty acids undergo B-oxidation inside peroxisomes, producing acetyl-CoA. The glyoxylate cycle then converts acetyl-CoA into intermediates that ultimately forms sucrose. This sucrose is transported throughout the young seedling, supplying the energy required for root growth, shoot elongation, and establishment. During the initial days of germination, seedlings cannot produce their own food because leaves have not yet developed and photosynthesis has not started. Therefore, all metabolic energy comes from stored reservoir inside the seed by enabling lipid breakdown and sugar production. Peroxisomes indirectly support ATP generation through respiration. Functional PX proteins ensure this energy supply remains uninterrupted, allowing vigorous and healthy seedling development. Reactive oxygen species or ROS are naturally produced during the intense metabolic activity of germination. In small amounts, ROS act as signaling molecules that promote germination and growth. However, excessive ROS can damage proteins, membranes, and DNA. Peroxisomes help maintain the proper balance of ROS, ensuring that beneficial signaling occurs without causing oxidative damage to the developing seedling. Catalase is one of the most important antioxidant enzymes found inside peroxisomes. Its main function is to rapidly convert harmful hydrogen peroxide into harmless water and oxygen. PX proteins are responsible for transporting catalase into peroxisomes. When this transport is disrupted, hydrogen peroxide accumulates inside cells, leading to oxidative stress, cellular injury, and reduced seedling vigor during germination. Peroxisomes also participate in the metabolism of important plant hormones. They contribute to the synthesis of jasmonic acid, influence auxin's metabolism, and are involved in pathways that regulate abscisic acid levels. These hormones control seed dormancy, germination, root development, and stress response. Therefore, healthy peroxisomes supported by functional PX proteins indirectly regulate hormonal balance and promote normal seedling establishment. Much of our understanding of PX proteins comes from studies on Arabidopsis mutants, plants carrying mutation in genes such as PX5, PX7, PX13, or PX14 show delayed germination, poor root growth, accumulation of unused lipids, and weak seedlings. These observations clearly demonstrate that efficient peroxisome, biogenesis, and protein import are essential for normal seed development and successful germination. Seedling establishment depends on the efficient utilization of stored food reserves. Functional peroxisomes convert stored lipids into sugars, regulate cellular metabolism, and protect cells from oxidative damage. As a result, seedlings develop stronger roots, longer hypocotyls, healthier shoots, and improved overall vigor. Proper functioning of PX proteins ultimately increases the chances of successful crop establishment in the field. Seeds often encounter environmental stresses such as drought, salinity, heat, or low temperatures during germination. These stresses increases the production of reactive oxygen species and reduce germination efficiency. By maintaining ROS balance and supporting metabolic adaptation, healthy peroxisomes help seedlings tolerate these adverse conditions. This makes PX proteins important not only for normal germination but also for stress resilience. Understanding the role of PX proteins has practical significance for agriculture. Improved peroxisome function can enhance seed vigor, promote faster germination, improve seedling establishment, and increase stress tolerance. These benefits are especially valuable in oilseed crops where lipid metabolism plays a central role. Knowledge of PX biology may also contribute to future breeding programs aimed at improving crop productivity and seed quality. Recent research is expanding our understanding of PEX proteins beyond their traditional functions. Scientists are exploring CRISPR-based editing of PEX genes, studying peroxisome signaling pathways, investigating interactions between peroxisomes and other organelles, and examining the role of PX proteins in seed longevity and stress tolerance. These advances may lead to innovative strategies for developing climate resilient crops with improved germination performance. To summarize, PX proteins are essential for the formation and maintenance of functional peroxisomes. They transport enzymes into the organelle, support B-oxidation, enable the glyoxylate cycle, regulate reactive oxygen species, and influence hormone metabolism. Together, these processes ensure efficient reservoir mobilization, continuous energy supply, and healthy seedling development. Without functional PX proteins, normal germination and early plant growth are severely affected. The key message of this video is that PX proteins are functional regulator of seed germination. By ensuring proper peroxisome functions, they allow stored lipids to be converted into sugars, maintain cellular redox balance, and support hormone regulation. These coordinated activities provide the energy and protection needed for vigorous seedling establishment. Continued research on PX proteins offers promising opportunities for improving seed quality, crop performance, and agriculture sustainability. Thank you for watching this video. I hope this session has helped you understand the important role of PX proteins during seed germination and their contribution to peroxisome function, energy metabolism, and seedling establishment. If you found this video useful, please like, share, and subscribe to Seed School India for more informative videos on seed science, plant breeding, biotechnology, and agriculture research. Thank you and see you in the next video.